All right, so welcome back. This is going to be the second recap video for this class. And I'll go over a little bit of review of what we just did. So a little bit of microscope work. Once again, I'll go through the anatomy of the microscope and a little bit of the best practices that you should be familiar with by now. I'll also go through a couple of the classes of Porifera, of our phylum Porifera. I'll go through what we saw on the microscopes when you were in lab Wednesday or Thursday. If I can change it, wrong way. There, for whatever reason. All right. So obviously, when you're using a microscope, carry it with 2 hands. If you don't, you have a higher risk of dropping it. And these microscopes are expensive, so you need to take care of them. So have one hand on the base, which is the structure right here, that bottom platform. Then have one arm or one hand on the arm. So there's this opening right here where you can hold the microscope. So when you're holding a microscope, one hand goes here on the arm, the other one's on the base, like that. So that's how you hold a microscope. When you put it down or you try to move it along a table, don't slide it. It has these rubber feet, which keep it in place so it's not sliding around. If you have to move it, pick it up and then move it. You risk damaging those feet, the lenses, and then the mirrors within the headpiece. So as a review, you look through these are the ocular lens. Light travels from this light source right here in the form of a light bulb. Light travels up through the iris diaphragm, through the stage, which is this flat platform here, onto the slide. And so the slide is what has our specimen, and that's what we're going to be looking at. From there, light travels up through these objective lens, and so those objective lens have 3 sets of glass inside. There's one that protects as almost like a dust cover. There's a piece of glass at the focal point, and so that's just this area where light intersects with each other. And then there's another piece of glass at the very top of that objective lens. So as light is traveling through that objective lens, it makes this X-shaped pattern. And so the cross in the middle of the X is where is what determines the magnification of that lens. So you'll have 4 different sets of objective lens, each of them with a different magnification, and each of those will have a different focal point, so the cross in that X. Light will continue to travel upwards into the headpiece, which is this structure right here, and then it will exit through the ocular lens, where we see it. When you're putting a slide on, you use the coarse focus to drop the slide all the way down. You put the objective lens in the lowest magnification, so that way you don't risk scratching the lens or the slide. You then bring the stage up so you can bring it into focus, and then you look through it, do what you need to do. When you're taking a slide off, you just reverse the process. So you take the magnification, you take it to the lowest magnification possible on the microscope, and then you drop the stage to the lowest it will go, and then you remove the slide. When you're using coarse and fine focus, you only use the fine focus knob, which is the outside, right here. You only use the fine focus knob when you're under high magnification, or you're trying to make slight adjustments to bring an object into focus. You use the coarse focus knob when you're trying to move the stage up and down really quickly, and you never use it when you have a high magnification. So if you do, you risk breaking the slide, so you'll have the objective lens, and then you raise the stage all the way up, and so the lens will be pressing on that slide. If given enough pressure, it will break the slide and possibly even the objective lens too. There's three different ways to control the amount of light that comes from the microscope. You have the iris diaphragm, which sits right underneath the stage. You can control the amount of light that comes in here. There's a small switch right here, so you can adjust the amount of light coming through. You can adjust the brightness by this light bulb. So there's a knob on the right side of the base, and it'll have a dim setting and a bright setting. There's another way you can move the actual iris diaphragm up and down instead of just closing that that light stage. So when you move the iris diaphragm up and down, you're focusing that light on that specimen. So that helps when you're under higher magnification. Focusing the light on a much more narrower point can make that specimen really stand out a lot better. Yeah, so that's microscopy in a couple minutes. So we went over sponge anatomy. We also did some work with trichonympha, but that exercise was really to get you all familiar with using a microscope and have some more practice. So microscopy is a great skill to have. I mean, I use it every day. I think everybody in the department considers microscopy as one of the most important skills you can have as a beginning biology student. So just going through trichonympha was a way to kind of increase the amount of practice you've all had with microscopes. We then went on to go through looking at a sponge. And so we went through looking at the spicules under the microscope. We also went through looking at a cross-section of that porifera. So what we should have seen was an osculum. So the very top part of the sponge, that hole right there, that's the osculum. Obviously, this is not the one that we saw on the microscope. This is just my favorite example. So we were able to see an osculum. We were able to see the ostia along the side. So there's openings to the spongicle that lead to a porocyte. So we take this example right here. You have this ostia right here, and then this is the porocyte, that opening. That leads to this central cavity that's called the spongicle. And then all these tiny lines in here are the spicules. These circles with a single line coming from it, those are the choanocytes. And there's pinacocytes that outline the individual. So when we looked at it under the microscope, this is what we saw. Obviously, it's a lot different than this one by a lot. So in this one, the most noticeable thing in here is this wide open cavity in the middle of the sponge. That's the spongicle. I didn't get a photo of it, but there's an osculum. So at the very top of this sponge, there's a very small point. Well, not small, but an opening here. That's the osculum. It's at the top of the sponge. There are pinacocytes. So if you look very closely, you can see the very thin cells that outline each of these buds. On the inside of those pinacocytes, there are choanocytes. So those are the flagellated cells that are undulating that flagella throughout the water, creating a water current, grabbing any food particles, and then bringing it towards them. You can't see any archaeocytes. Those have been long gone. You can't see sclerocytes. And you can't see spicules. So when they make these slides, they take a really sharp knife, they cut through the porifera of the sponge, and when they do that, all the spicules shatter. So they've been washed away, and we can't see them here. There's an ostia, so the opening of a porocyte. Let's see if I can find one on this picture. Well, if you look closely enough, you'll find one. basically, these areas here are porocytes. So there's one here. There's another porocyte here, 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 so on and so forth. Here's a really good one right here. And so when they make these slides, they take that sharp knife and they just cut one individual into multiple cross sections. So the chances of us having a very clear ostia are rather slim. So, but like I said, you can see the pinacocytes, the outer layer of that that porocyte right here. You can see the spongiocele. You can see the osculum, just the very top part of that cavity. What else can you see? You can see the choanocytes. So these dots that are all along the inside of this bud. Yeah, that's really all you can see. It's not as good as the diagrams. So this is what the spicules look like under the microscope. I like to say they look like a Mercedes-Benz symbol or a peace sign. Choose your pick. So when they make these slides, they just take a sponge, they dip it in acid until all the organic material wastes away, and you're left with mesohele, that acellular matrix, and then these spicules. So this is what's left over. And so when these, if you saw spicules in a regular living sponge, they'd be all interlaced together, and then that mesohele would be filling in any gaps between them. So yeah, there are the three classes that we went over, and they're all based on what is the chemical composition of those spicules. So we have Calciospongiae, obviously, or not obviously, but they're the easiest to remember. So they have spicules that are made of calcium carbonate, and so hence the name Calciospongiae. The second easiest, I think, to remember are the Hyalospongiae, the glass sponges. So they have their spicules formed out of silica and only silica. And so silica is what glass is made out of. And so they have these really clear and really intricate spicules. So there's the one specimen already set in the back, but it looks like it's just this this 3D printed net of this translucent glass material. And so that's Hyalospongiae. I think that's the second easiest to remember. The third class is Demospongiae. That's this stuff like this, like bath sponges. And so those have spicules made out of spongin and silica. So that's a review of what we did this week in lab. Next week, well, I guess Sunday, I'll go through a lecture talking about the phylum Cnidaria. So hydra, sea anemones, corals, and jellyfish. And then I'll also go into the phylum Platyhelminthes. So these are the flatworms. We'll play around with some planaria. We'll go through some of the more disgusting organisms that we talk about, such as tapeworms, nematodes. Actually, not nematodes. We don't talk about those yet. So these two groups both have a really major step in evolution that we cover. And so one of that is we now have true tissues. So the difference between Porifera and Cnidaria is that Porifera lacked true tissues, while Cnidaria actually has tissues. So Porifera only had specialized cells that were working together. Cnidaria actually has these specialized cells working together as a single unit. In the phylum Platyhelminthes, we have true tissues, but instead of having two sets of internal body tissues while developing, we have three, and then we have our first bilateral organism. So instead of having a round organism where you can cut it in any way you want and have the same thing along the axis, we now have an organism that you can only cut it along the length of its body for it to be symmetrical. So both of those are rather important evolutionary steps. And I'll talk about that a lot more well, Sunday. Until then, continue to wear your masks. Yeah, have fun. Not too much fun. Wear your mask.